Good morning and welcome. Thank you for joining us for Myrtle's second annual industry-sponsored symposium. I am Jordana Holovac, Head of Communications and Community at Myrtle, but my badge of honor is that I am a rare disease mom who became intimately involved with gene therapy when my son Jacob was diagnosed with Canavan disease in 1996 and became one of the first recipients of gene therapy for a brain-based disorder in 1998 at Yale New Haven Children's Hospital. It has been a quarter of a century since Jacob's first treatment, which utilized a liposomal vector. It is amazing to think that as that vector was being delivered into his brain, we were already looking ahead and planning for future treatments that would include an AAV delivery, which he received a couple of years later. The field has come so far, and the promise of treatments never more exciting. So you can only imagine my excitement and pride to be able to coordinate symposiums such as these, where we have the opportunity to take a deep dive into the emerging role of oligodendrocytes, these amazing myelin-making cells that play such a critical role in white matter diseases and myelin-based conditions, and perhaps a broader role in cognitive function, such as memory and learning. With that in mind, we have brought together a team of experts who have focused on oligodendrocytes throughout their work in non-clinical development, as principal investigator on Myrtle's oligodendrocyte-targeted gene therapy in Canavan disease, and in the discovery of novel oligodendrocyte-targeted AAV capsids. It is important to note that Dr. Lober was unable to be here this morning due to a patient emergency. But Olga Flamini, Myrtle's medical director, will deliver his presentation and is certainly up for the task. She is a critical part of the clinical team at Myrtle. Given our sensitivity to the clock, I will introduce each speaker as we go through each session one by one. And as a point of order, we will use our time today for presenting and will not have a formal Q&A. However, we will have a bit of time following the presentations to gather informally for introductions and one-on-one questions as the room is being prepared for the next group. It is my honor to introduce our first speaker, Dr. Paola Leone, professor and director of the Cell and Gene Therapy Center at Rowan Virtuous School of Osteopathic Medicine and School of Translational Biomedical Engineering and Sciences at Virtua Health College of Medicine and Life Sciences of Rowan University. Paola?
To this symposium. Thank you everyone for attending today. I'm not used to podium. I mean, I am, but I much more prefer to be looking at you when I present. So in this presentation, we'll recapitulate 25 years of work that we have conducted in Canavan disease in terms of preclinical and also clinical data. And so this is the exciting part that we have. We can really discuss the value of preclinical studies in animal models or in animals versus comparison with clinical trials that I conducted in patients as IND-sponsor clinical trials. So
It's not going to put up. It's not working.
Sorry. Okay. First of all, disclosures. As important to note that I'm currently a consultant of Myrtelle Biotherapeutics. I'm also a founder. Since this project started from the lab, really from my bench in collaboration with academic investigators, and then developed into the amazing program that is conducted at Myrtelle. So we'll be discussing today just a couple of things that are important to emphasize in this disease. So the genetics, the biochemistry, and of course the neuropathology and clinical features of the disease, as well as the history of Canavan clinical gene therapy in Canavan, and what we have learned from this, and how we then decided to focus on the best, most natural way of developing the treatment for this disease, targeting the natural compartment where...
Paola, come to the podium. No one can.
Sorry about this. Back to the podium. Okay. And so what we'll be discussing today, aside from the neuropathology, is also the clinical gene therapy program, so 25 years of research in patients with prospective treatments, as well as a collection on natural history of this disorder. We'll discuss animal models. Animal models are very relevant when we want to determine justification for studies in larger animals as well as in patients. And then some of the data with the Canavan disease biochemical mechanism, as I emphasize, the mutation in the aspartate gene, which is primarily present in oligodendrocytes, results in the inability of NAA to be hydrolyzed, and hence the result is elevated NAA and lack of acetyl group, which are really promoters of myelin. And this is also a diagnostic, very simple for patients that may be diagnosed with leukodystrophy. Urinary acid analysis is a quick test that can be done, and NAA in urine spike up as well as in the brain. Very important, NAA is the second most abundant derivative amino acid in the brain, which role is, aside from the role of providing acetyl group, is not well understood, still unclear. So I thought we would just present an animation here, so to clarify where the function of the normal aspartate activity in oligodendrocytes. NAA just enter oligodendrocytes and then is broken down in acetyl group that are just necessary to promote myelin. The result of the mutation, as is clarified here, is that there is a buildup of NAA with a reduction or no acetyl group that, in fact, transfer to the axon. There is a demyelination, and nerve impulse is blocked. So to give a visual representation of a case study in Canavan disease, here's a 10-month. We have an axial T1 weighted image in a patient with progressive degeneration that first results in that demyelination, and then severe atrophy at the age, in this case, 64. So the clinical feature of the disease is that the patient presents with general weakness, inability to meet any significant milestones, poor head control, poor trunk support, first floppiness, and then hyperflexia, seizures, macrocephaly, and everything associated with this type of condition, which, as you can see from this sagittal MRI from the same patient at 2 and 5 years, shows initial demyelination at 2 years of age, and macroscopically you will already see vacuolation in the brain throughout cingulum cortex, pons, and cerebellum, with progressive brain atrophy. I will see it here at 5 years of age. The neuropathology is very clear, with brain edema and really disintegrated myelin and brain atrophy. The history of the clinical Canavan gene therapy is very important because we learn a lot from our experience. We started in 1996, and that was 2 years after the discovery of the gene, aspartate, and we were able to have approval for a non-viral vector, a liposomal vector, which we introduced in the cerebrospinal fluid using an Ommaya reservoir. And those were early studies, were funded solely by foundations, and we published a portion of the data later, and as soon as we were already working with AAV, we already published in an animal model of Parkinson's disease the effectiveness of raising tyrosine hydroxylase, a modifying disease in rats. So we were working with AAV. We couldn't use it in the clinic because at that time we needed to have a wild-type adenovirus for the packaging of the vector. And then when the seminal work of Samolsky and Zhao Zhao was published in 1998, and we were able to promote work and use a triple transfection without the use of wild-type adenovirus, we were good to go. We were good to go, and there was early in 1998, 1999, preclinical data, safety data, large animal model, large animals that we used for the safety, and eventually the NIH and the FDA approved NIH for funding and FDA for clinical trial. The first application of AAV into the brain in June 2001, and that's where we started with the first 3 patients, and we continued the work that was funded by the NIH, in which we also collected consistent natural history data in patients that were not treated longitudinally, and that study focused on the NAA being synthesized primarily in neurons, and so we targeted neurons because we want to reduce the NAA at the source. That was the idea. So it was neurotropic and was intraparenchymal, 6 injections delivered in the parenchyma that then would be followed through single voxel NMR spectroscopy for absolute quantitation of NAA and other metabolites in sites of injected sites in the brain. So this study was very important because when we completed it, it was clear to us that we did, these are the data presented here were published in Neuropediatrics. Just to give you highlights, we are presenting ADC data, diffusion, apparent diffusion coefficient, which is a marker of, neuroradiologic marker of myelin and lipid function in the brain. We do know that just like as per brain atrophy, there is a reduction of ADC in the brain of patients. There are 2 areas highlighted here. There are many more in the original article. And so it was clear to us that, of course, the demyelination is also followed by brain atrophy. And the result of the study showed that we did indeed reduce NAA, specifically in many areas and significantly in periventricular and frontal area. However, this reduction in NAA was not associated with myelination. And when the youngest patients came along and with a team of investigators, we were looking at them, that's when we completed the study, collected the data, we published this work, but I knew that what we had to do was to detarget the expression of ASPA in the natural compartment in oligodendrocytes. Also because during those days and during those time since the AAV1 application, several Kahneman models were developed, very important to select the correct Kahneman model. 2, the tremor rat is a spontaneous mutation in the aspartoacylase, deletion of the aspartoacylase gene with many other genes. And so it's not the cleanest model because there are other genes involved. There is also what we call the Matalon mouse, which is called knockout in literature, but is a knock-in of a deletion, so knock-in which result in knockout. And the lifespan of that mouse, as published, is variable from 21 days to several months. And so because of that, well, first of all, that model is not publicly available, but also if I had to choose, even if are publicly available, I would not choose a model with a lifespan that is so variable, because that would may lead to a type 1 error statistic. So obviously, because there is a variability in lifespan. So we chose the most characterized animal model, the NUR7, which replicates all of the pathology and motor dysfunction of Kahneman disease and has a very stable lifespan of 10 months. And this is all the work that we have done to determine proof of concept was in fact done in this animal model. So our preclinical studies started in, as always, as a proof of concept in pre-symptomatic mice, P1, P2, just newborn that were injected in several brain areas, cingulum, cortex, cerebellum, so intraparenchymal. And the first point was targeting was the AAV olig, which was developed at UNC by Steve Gray, and was shared with me through an academic material transfer agreement. was used in this animal model to determine the tropism, and in fact, the tropism was very clearly, as shown, was preferential for oligodendrocytes. And so we designed a codon-optimized cDNA that was functional. In this case, we have GFP in the panels that you see expressed with co-labeling for several markers, and OLIG2 is the one with the red column that shows primary tropism. In the paper that is published, we show that we were able to rescue the metabolic deficits associated with Canavan disease. Well-established elevation in NAA was reduced, as well as increase in ATP and oxidative markers that are characteristic in this disease, in this model. And we also resolve the vacuolation and the motor deficit, which, as we can see here in panel A, we are showing the black column shows AAV olig versus the AAV GFP, the AAV olig GFP versus the ASPA, and the difference in the effect. While the panel B shows the total locomotor activity during a day. And so there was an increase in locomotor activity, and we can see that the column, white column versus black column, are indistinguishable. So we brought our NER7 animals to total functional recovery. But then we had to move on into the clinic. Patients are not typically diagnosed at birth. They are diagnosed within the 6-10 months first year of life. And we needed to develop a program that would really address patients when they are diagnosed. And so the symptomatic patients, the comparability was at 6 weeks in the NER7 mice, which is during that time total development of myelin, as published by many groups, which really recapitulate the phenotype of a patient diagnosed of a child with Canavan disease at 24 to 30 months. We initially looked for biodistribution studies to the best route of administration, which in the study published show to be the yellow columns in the panel show dose response of the AAV olig GFP through the ventricles. So that we selected the ventricles, the ICV route, as best route of administration. And in addition to that, in a wild-type mouse, we looked at the tropism of two different vectors, the AAV olig and the other most widely used, AAV9, showing that these are striatal injection of the vector, which show tropism 80%, about 75% of a oligotropic tropism of the AAV olig versus the AAV9, which shows 70% GFAP, so astrocytic tropism. In addition to this, also published is the fact that treatment with AAV olig ASPA resolved at age 6 weeks, so post-myelination, resolved the vacuolation in the brain at two different doses. The highest dose of AAV9 and AAV olig result indistinguishable. You see the first column. And we have to remember the tropism and the fact that to have the same effect of AAV9 to AAV olig, we need a 10 times more dose to be applied. And so in addition to this, very important data is a phenotypic correction of the motor function. And so we have, in all treated animals, we have the olig, a superior effect at every dose, especially the higher dose as compared to AAV9, as you can see in these graphs. So the conclusion is that we selected the AAV olig as the most appropriate vector for this disease. The route of administration, well established, ICV, intracerebral ventricular. The findings supported the application. These findings supported the application of AAV olig in the clinic, and so this is like how we would like to see just the development of the disease in a Kahneman brain, so just myelination after the aspartate expression, and we'll see data that support this idea and this efficacy. Of course, acknowledgements, first and foremost, families and patients, patients advocate, Jordana Olovak here, who just for 25 years supported program and development for her son and many other children diagnosed with this disease. Special thanks to Rob Lober and the clinical team in Dayton. They are amazing and compassionate, and in fact, the reason why Dr. Lober couldn't be here is spend the night in the OR caring for a patient for an urgency. So also very thankful to the Myrtelle founder and all of the people that just supported the program, which is now in phase 1, phase 2. And Jeremy Francis in the lab, who just generated supported and managed most of the preclinical studies. And at this point, I would like to introduce Dr. Olga Flamini, that will be presenting the next presentation on a case report in Kahneman disease. And Olga started with me from day one since the 2019, 2020. And she's a medical director at Myrtelle and acting CMO.
Good morning, everyone. Good morning. Thank you for coming. So, as you can see, I'm not a Rob. And today we will talk about the progress in the ongoing phase 1/2 clinical trial in patients with Kahneman disease under Myrtelle sponsorship. Kahneman disease was first described in 1931 by Dr. Myrtelle Kahneman, who was a neuropathologist in Boston. And she observed the spongiform degenerations in the brain of patients affected by rare disease. Later, this disorder was named Kahneman disease. The company I'm representing, Myrtelle, was named as a tribute to Dr. Myrtelle Kahneman and her discovery. Today we know that Kahneman disease is a rare genetic neurodegenerative disorder which results from the mutation of ASPA gene responsible for aspartate enzyme production. Normally, aspartate breaks down N-acetyl aspartate into amino acids acetate and aspartate. As it was covered during the previous presentation, oligodendrocytes is the main compartment where aspartate is normally expressed. Therefore, we use an oligodendrocyte-specific vector to deliver a healthy copy of ASPA gene to oligodendrocytes, where it is needed the most for the myelination. We think it will help to restore the oligodendrocyte health and myelination, and will help to improve patients' functions in the future. Today, so far, totally 10 patients received MYR-101 gene therapy, and in this presentation, I'll talk in more details about first 9 dosed patients, since the last patient was dosed just last week after the presentation cutoff. All the patients totally follow up for 5 years post-treatment and receive intracerebral ventricular injections of MYR-101 in lateral ventricles. We conduct different tests to assess the safety and efficacy of the drug. Patients who signed informed consent and were found eligible based on the inclusion and exclusion criteria were enrolled into the study. Inclusion criteria included definitive diagnosis of Kahneman disease, aged between 3 and 60 months at the time of gene therapy, and neuroimaging within 3 months for neurosurgical planning. Exclusion criteria included significant medical conditions, making the patients not a suitable candidate for participation, history of severe allergic reactions, previous gene therapy, prior intracranial surgery, contraindications to immunosuppressions, life expectancy less than 12 months for any reason, and GMFM raw score more than 35%. On this slide, you can see demographics for first 9 dosed patients. And right now, we are preparing a manuscript for publication of this patient's 12 months data. And to avoid the conflict with publishing, we chose to present a single patient case report. All the patients were diagnosed with neonatal infantile form of Canavan disease and were severely affected. But despite this, this is a heterogeneous patient population, and it was hard to make a decision to choose the average patient to present. Therefore, we chose to present patient number 6, who had sort of average, but a little bit, he was younger than average age, and he had a little bit higher functioning, higher motor function compared to other patients, though his cognitive function was comparable or lower to the rest of the patient. This patient met all inclusion criteria and had 2 pathogenic mutations in ASPA gene. Also, this patient had biochemical and neuroradiology changes characteristic to Canavan disease. By 6 months, microcephaly was noted in the patient, and by 12 months, patient plateaued, and after 12 months, patient did not gain any significant milestones. Patient underwent neurosurgical procedure where temporary catheters were injected in the frontal horns of the lateral ventricles. Some amount of CSF was removed prior to drug injections, and the patient received the total injected dose of 3.7 to the 10 to the 13 viral genomes in lateral ventricles. Surgery was uneventful and lasted about 3 hours, and patient was discharged 3 days later. Regarding the safety, this patient had no adverse events associated with either drug product or neurosurgical procedure. And now to the exciting part, the efficacy results. To assess neuroradiology changes, we use synthetic MRI technique. Synthetic MRI is an advanced imaging technique which enables precise quantification of different brain tissues by differentiating and quantifying between various brain tissues, like gray matter, white matter, myelin, water content. And on the figure on the top, you see a myelin development in the healthy child from 1 month to 15 years. And myelin here you can see in different shades of green and yellow, and intensity of the color indicates the higher myelin levels. On the figure below, the line, the solid line, represents the average amount of myelin across the age. And you can see that the red dots overlaid on the figure represent the patient myelin levels, and you can see it's on the lower levels compared to age comparators. So by 12 months after the treatment, we observed significant improvement in both myelin and white matter levels, which changes the natural course of the disease progression. And this improvement is a dramatic improvement never described in the literature in Canavan disease patient. To assess the function of this patient, we use tests so-called Merlin scales of early learning, which allows to assess the patient performance in different developmental areas, like it allows to assess gross motor function, fine motor function, receptive language, expressive language, visual reception, and we noted an improvement in the most areas according to this test, and also in 5-domain mean, which sort of represents the average developmental age of the child based on the different areas of development. To assess specifically gross motor function of the patient, we use tests called gross motor function measure. And we noted improvement in such domains as laying, rolling, sitting, and crawling and kneeling. Prior to the gene therapy, patient had abnormal muscle weakness, lack of truncal control, and very weak head control. 12 months after gene therapy, patient is able to sit without support, interact with toys, and even attempts to take steps. So, sum it up, we would like to acknowledge contribution and effort of everyone involved in the clinical program and extend special thanks to patients and participated families and site staff at Dayton Children's Hospital. Thank you.
Thank you, Olga. Our last speaker this morning is Patrick Aldrin Kirk, Chief Science Officer at Raven, an AAV engineering company focused on generating innovative vectors for the next generation of gene therapies. Patrick?
Thank you for that introduction. So my name is Patrick Aldrin. I'm the CSO at Raven Therapeutics. And as we all know, oligodendrocytes are now implicated in hosts of several different disorders and are now being actively targeted as gene therapy approaches. So I think Myrtelle has been really pioneering in developing these AAV-based gene therapies. And I also think this highlights the continuous need within our field to refine and enhance the efficacy of our AAV vectors. So before we go into that, I would like to talk a little bit about our company, Raven Therapeutics, which stands for Recombinant Adeno-Associated Virus Engineering. We were founded in 2022 in Lund, Sweden, by our current COO, Marcus Davidson. And we really focus on capsid engineering for increasing the cell and tissue specificity of our vectors, while also detargeting that capsid to other tissue types or cell types. In addition to that, we try and engineer our vectors to take advantage of endogenous transport mechanisms to really make sure that our vector can reach those cells that we wish to target. In addition to that, we also target the capsid engineering towards avoiding the immune response, as well as combining novel promoter and enhancer screens in order to improve the specificity of the transgene still further. So we focus on peptide fragment insertion in the AAV capsid to achieve this. And we do this because we think there are some limitations to the current AAV wild-type vectors. And this includes low specificity against several different organ types and cell types, as well as low infectivity of those cell types, which then also requires a large dose of virus. And this presents the challenge out of a safety aspect when you have to go into and utilize a very large dose of virus in patients. We have also shied away from the approach of directed evolution because we feel that this is a costly and time-consuming approach which requires multiple screens. We also believe that this approach may result in the risk of poor translatability, since this is an entirely stochastic approach where you don't really target any specific viral entry mechanisms that are conserved between your mammalian species model organism and your, viral entry mechanism present in patients. So this is where we come in, and we address these limitations by a rational designed approach where we look at the receptors that are present on our target cells that may be viable viral entry receptors and target our capsids against those. So this is a very brief workflow of how we operate. So we first start with targeting the tissue or cell type and identify the receptors that are present on those cell types. We can then engineer a large library designed to interact with those specific receptors to gain entry into those cells with our viral vector capsids. Another important aspect of our approach is the utilization of DNA barcodes. And a DNA barcode is essentially a small stretch of nucleotides that help us identify the exact capsid that was able to enter our target cells. And a short stretch of 20 nucleotides allows us to label up to 1 trillion variants in each screen. Right. So we start out with first cloning in our peptide fragment into the cap gene of our capsid. Simultaneously, we also clone in the DNA barcode into the transgene expression cassette. And this allows us to link the peptide insert fragment to the DNA barcode. So when we move and pull out those specific cells that we're interested in targeting, we can sequence that DNA barcode, and that will contain all the information we need to know exactly which peptide was present on the AAV capsid that allowed for that cell to be infected. So I was told there was going to be a delay here.
Oh, there we go.
Okay, great. So I want to highlight a couple of vectors and capsids we have developed to date here. We can start out with a library that we designed to spread and diffuse throughout the entire tissue through a single injection. I think you can appreciate the difference here between a standard AAV2 single injection into the brain parenchyma versus our designed library. And you can keep this library that we designed in mind when we compare that to the library that we have designed to target oligodendrocytes. We have also targeted synaptics capsids that are able to infect synapses and are then able to be retrogradely transported through the axon back to the cell body. And this is where we also highlighted that we can use the exact same capsid with against the exact same target receptor, both in the rat neurons as well as human transplanted neurons with a similar efficacy. Of course, we're also trying to enhance our vector's efficiency. And on the last comparison here, you can see even at low titers, when we screen vectors for increased efficiency, and this is in culture on human neurons, that we are outperforming the standard AAV2, AAV8, and AAV9 in this case. So to sum up what our approach, we think it's a fast approach that requires only a single screen. We also think this is more translatable because we are targeting very specific viral entry mechanisms that are conserved between both the mammalian model organism and the patient. We're not limited to any specific model system. When we do these screens, we can screen in anything from rats to NHPs. And the beautiful part of our approach here is also that we can target several different cell types in a single library screen and look for different capsids for different target cells. And this has drastically allowed us to reduce the animals that we need for every single screen. And taken together, I think this is a more cost-effective and lean approach to developing novel AAV capsids. So onward now to the oligodendrocyte targeting. That is in our collaboration with Myrtelle. So in this case, we started out with identifying oligodendrocytes and the viral entry receptors present on oligodendrocytes. We designed a library of viruses that could interact with these receptors and then engineered those and injected that library into our model, which is in this case rats. So as an example, we would identify MOG as a possible viral entry receptor, and we would then derive our peptides from other viruses, other microorganisms, endogenous proteins and peptides that could interact with the MOG receptor for viral entry. So this is the workflow in this screen, which we call the Kingfisher screen. And we first injected our library into the parenchyma of rat brains, and this was done just to get access to a large amount of transduced cells that we could then sort for GFP and for SOX10, which is a marker for oligodendrocytes, as well as sort those cells that were SOX10 negative. And this would give us two populations where we could compare the capsid's performance in both targeting oligodendrocytes and non-oligodendrocytes. We also injected the library into the ventricle, and this was done to also screen for a capsid that is able to become transported from the ventricle into the surrounding parenchyma. And here we just collected biopsies throughout the brain tissue to assess this, focusing on the white matter tissue. So when we combine the sequencing results from both these approaches, we're screening for a capsid that is specific for infecting oligodendrocytes, as well as has the capacity to become transported from the ventricle and into the surrounding brain. And this allowed us to then make a short list of capsid candidates to evaluate. So this is an example of our library injected into direct injection into parenchyma. And if you remember the earlier library that we designed to spread throughout the entire tissue, this library is far more targeted towards the white matter tissue. Not only that, but it also spreads from the injection site into a host of other white matter tissues. Similarly, when we injected into the ventricle with our library, we saw an enrichment of GFP-positive infected cells in the white matter regions that are adjacent to the ventricle, and in this case, such as the stria medullaris and posterior commissure. Of course, we also wanted to verify that the cells that we are targeting, or the library that we have developed, can also target oligodendrocytes. And in this case, when we injected the virus into the corpus callosum, we also see a broad overlap between expression of GFP and SOX10. So we're very confident that we're able, in this library, to target oligodendrocytes. So when we do a comparison based on the sequencing and the amount of transcripts that we find based on the barcodes that correspond to different capsids, we can make a ratio how often that capsid is found in either oligodendrocytes or non-oligodendrocytes. If we then also plug in the sequencing results from the intraventricular injection, we can sort of tease out which vectors are our main candidates that have both the ability to infect oligodendrocytes, as well as have the ability to be transported from the ventricle and into the brain. So I'm going to end here with just one example of one of our capsids that came up as a candidate, which we call the Kingfisher 10. And I think this is the immediate injection area in this case. And I think you can appreciate immediately that you don't really see any cells here that are GFP positive that have any neuronal morphology. And when we also look closer, we can also see that a lot of these cells are actually SOX10 positive, indicating that we are indeed targeting oligodendrocytes specifically with our approach. I would like to highlight here that this was done in a single screen, and that we are in this validation study using a CMV promoter. So, and I think this really showcases the power behind our approach to developing novel AAV capsids. So with that, I would like to thank you for listening, and I would also like to thank Myrtelle for inviting us to present at this symposium. So thank you very much.
Well, look at us, so efficient. We actually do have some time for Q&A. We have about 15 minutes, and then, of course, we can congregate a bit for some more informal discussion as they're preparing the room for their next speakers. So we can open up the floor up. Oh, I don't know if it's on. Oh,
I'll just yell really loud. So,
sorry.
congrats to the team. The data looks really fantastic, so I'm really excited to see that. I have a question for Patrick, though. So, the capsid, the approach, if I'm understanding it correctly, is to put in already known peptides and other things that you think would interact with a particular cell type, not a random screen. Is that correct?
Yeah, that's correct.
Oh.
So we are actually looking at the specific domains of that receptor and looking at which other domains from other sources, such as other viruses or microorganisms or endogenous proteins that are actually interacting with that binding pocket, and base our screen on that.
Okay, so I'm curious. Oh, nice. So I'm curious, in following up now that you have found some capsids, have you been able to demonstrate that any of these capsids are actually working through the receptor that you designed them to work through? And sorry, and or if there may be a possibility that you've actually enriched or selected for ability to be transported along axon tracks. Excuse me.
So
It looked like you saw a really nice track.
Yeah. So in this particular screen, we haven't done that study as of yet, but we have done this in our previous studies. One example is when we did a targeting towards the synapses, which I showed. We were actually using tau and that receptor of tau. So we were able to block entry with our capsid with addition of tau in that case. And we were able to block the synaptic infection in that case.
Any other questions? Yeah. You want to go to them? Yeah.
Thank you very much. A great number, actually. So one of the things I'm interested in is what the mechanism of transport is, do you think, you're diffusing initially, you're getting into oligodendrocytes, but, with axonal transport, it's fairly straightforward. You can understand the mechanism. To get wide distribution of oligodendrocytes, unless it's axonal transport, is a puzzle to me.
So I guess the question is for me. Or, yeah.
Sorry, Patrick.
Yeah. So in this case, we're also doing a little bit of other targeting as well. So we would, in this case, target receptors that are able to do receptor-mediated transcytosis through the ependymal layer to then have that ability. And then we can combine the availability of that approach when modifying the capsid with the approach of also targeting the oligodendrocytes.
And then will that get you the distribution you need over these quite big distances?
Yeah, so this is something we're currently assessing right now. The last image I showed was taken last week, just before we flew here. So that's an ongoing study right now.
Great.
Yep.
Thank you very much.
Hi, great work. And I had a question I've always wanted to ask about your imaging method, the synthetic MRI. Not as a radiologist, is there a sort of soundbite explanation of how that's done? And then the distinction between white matter and myelin quantification that you've shown. What's different about those and how this is captured or analyzed?
Thank you. Thank you, Jenny, for a good question. Synthetic MRI done in parallel with the traditional MRI and method, and it uses the the magic sequence, and the quantification happens afterwards on the analysis, post image analysis level. Regarding the difference between myelin and white matter also includes the axons, when myelin is just the myelin. That's it.
Hey there, Andrew Steinzepper from Deerfield Management. The data was amazing to look at, and congratulations for all that work and those patient volunteers that are there. Now, given where you are, and with everything you know now, thinking about the next indication and the next efforts, what would you be changing in this approach? Because I know this took some time to get there, but especially for folks out here that are trying to develop novel efforts and start over or start things new, what lessons learned would you want to make sure, those hard-won lessons that everyone else is taking from the work that you've already done?
Everything we do is obviously a work in progress, and this is like a really good question. If we could go back 20 years ago or 5 years ago, what would we do? We would probably look at a dose response curve, so higher doses, and perhaps we would also look at additional route of administration. You mentioned indications, so we have Canavan disease, and in Canavan disease, really, the synthesis of NAA is primarily in neurons, and so by directed diffusion osmosis from the brain to the CSF, to blood, to urine. So if we hypothetically place another kidney, so the idea would be to lower NAA, but that lowering NAA throughout the CSF and the brain CSF and urine would not result in myelination. So, however, probably would improve the issue related with edema in this patient and macrocephaly, which is very visible at diagnosis. And so studying the disease in patients and collecting really valuable natural history is what I would do all over again. And of course, we learn about the targeted therapy to neurons. Of course, there are two compartments. We're targeting NAA because that's what we knew, and then we realized that naturally, aspartate in oligodendrocytes, and so that was the best. And we didn't have an AAV in those days. That the first one was the one that Steve Gray developed, and so we jumped immediately into it, and that's where we are. And so any other technology that's just developing for additionally oligodendrocyte AAV, I think is very relevant. But overall, I would say while the program is moving ahead, we're going to be able to build parallel lines of exploration in which perhaps we'll look at just different variables like dosing, and with all the programs with the FDA and expanded access, we could design studies like that, and also include perhaps older patients and maybe even younger patients, as now they're becoming available. So there is a lot of work in progress, and we're all prepared for it after so many years of working in this disease. But thank you for your question.
Fantastic data on all fronts. I had one more question on the AAV engineering side of things. It's a super cool approach, and I was curious sort of on the choice of the target receptor, and sort of just like what led you to a particular receptor, if there's any ones where it seemed like it would be cell type specific, but it worked very poorly as a target for actually getting AAV transduction, just kind of general lessons learned there.
Yeah, so the example that I showed was just an example of a receptor, not saying that we actually targeted that specific one, but we are mainly targeting receptors that can be used for internalization. So receptors that may internalize antibodies or other virus capsids or something to that extent. If it's not really an internalizing receptor, we don't really utilize it. And then, I mean, of course, there are other aspects we take into account as well, if it goes into an early endosome or such things as well. That's how we essentially choose our targets.
Interesting. And have the results sort of generally borne out with intuition there for what would make a good target or not, or have there been wildcards?
There have been wildcards as well, of course. But in general, I would say we screen for the targets. We're successful in screening for successful capsids in the target we have tried to do so far, at least. We've not really failed as of yet in finding a capsid that doesn't work for a specific target. So we kind of like this approach.
Very cool stuff, yeah.
Yep.
Thank you, everyone.
Thanks.
Hi. Wonderful talks. Thank you so much for sharing 12-month post-treatment video for patient 6. I know you said there's a paper coming, but I was wondering if you could give us a little bit of a preview. Where's patient 6 when you look at the overall response in your trial? Is that typical, atypical, somewhere there?
Thank you for question. Regarding the state of the publication, we are preparing the manuscript currently. And as I mentioned, patient's baseline was, from motor standpoint, he had a higher baseline compared to other patients. His cognitive baseline was comparable or lower to the rest of the patients. His average age, but on the younger side. We think he's one of the better performing patients in a trial, and we have lovely videos for him. That's why we chose to present him today. But overall, I would say he was a severely affected patient, and his improvement is comparable to the rest of the patients in comparable age group we dosed.
Thank you all for joining us this morning. I'm sorry we have a hard stop, but like I said, we can congregate a little bit informally while they're preparing the room. But thank you all, and enjoy the conference.